This model represents a cutaway view of a synaptic vesicle, with the membrane lipids and proteins drawn to scale. Each synaptic vesicle membrane contains approximately 7,000 phospholipid molecules and 5,700 cholesterol molecules. Each vesicle also contains close to 50 different integral membrane protein molecules, which vary widely in their relative abundance. The most abundant protein is a snare protein, called V-snare. This molecule participates in membrane fusion at the synaptic terminal, and there are about 70 copies of this protein per vesicle. By contrast, the vesicle contains only 1 to 2 copies of V-ATPase. The V-ATPase uses energy from ATP hydrolysis to pump protons into the vesicle lumen. The resulting electrochemical gradient provides the energy to import neurotransmitter molecules, such as glutamate, into the vesicle. Through a transporter in the membrane, protons flow down their concentration gradient to the outside of the vesicle, as glutamate enters by an antiport mechanism. In this way, a vesicle is loaded with thousands of glutamate molecules. This model was created in part from an electron tomogram of a real synaptic vesicle. The tomogram combines visual slices of the vesicle at various angles to create a unified image. Other data, including structural data of the synaptic vesicle's proteins, have been combined with the data from the tomogram to create the three-dimensional model on the left. Note, only 70% of the membrane proteins estimated to be present in the membrane are depicted in the model. A real vesicle would be covered by an even more dense forest of proteins.